Hello everyone. Welcome to part 7 of the lecture series. So this lecture is extremely important because this is on protein structure and recently a lot of research has been going on here. I mean, if you have heard about DeepMind and AlphaFold, you also know about this. So let's dive deep into this. So the first thing is we know that a protein sequence it consists of 20 different amino acids, right? So it's basically a combination of these letters. That is the very fundamental thing about proteins. Now, the primary structure of protein, which is the most fundamental structure, it is nothing but a sequence of amino acids. So just to make it clearer, so you have a lot of amino acids joined together using a bond called peptide bond. So this sequence, it's also called polypeptide chain. So it's a chain of amino acids. And another important thing is there are two terminals here. The protein sequence or the polypeptide chain starts from the N terminal and it ends in the C terminal because the starting radical is NH2 and the ending one is COOH. That is why. So previously in DNA we talked about 5' and 3' end. So in proteins we are going to say N terminal and C terminal. Okay. Now a polypeptide chain or a protein can be moderately long, I would say. It can have hundreds or even thousands of amino acids. Now, amino acids which remain inside of the protein or the is part of the polypeptide chain is also known as residues. So in many papers, we're going to see this name as well. So this is the primary structure of the protein. Now, this picture we have seen now, I think 3 times in our previous lectures. So the point I'm trying to make here is if this is a gene and there are a few exons in this gene, then through alternative splicing, we can create multiple mRNAs or messenger RNAs from this particular gene, right? And each of these mRNA will translate into a unique protein. So what I'm trying to say here is that one single gene can actually create multiple proteins. And that is why the number of proteins in the real world is much greater than the number of genes. I mean, we have a lot of genes. We have around 20,000 or even more protein coding genes, but still the number of protein is much greater than 20,000. So next, let's talk about the secondary structure of protein. So primary structure and secondary structure. So secondary structure depends on the different chemical bonds between the peptide and the amino acid. So it's an interaction type of thing. So we have different types of bonds such as hydrogen bonds, pi bonds, and there is the amino acid and peptide chain reaction. Now, depending on how these bonds are actually occurring, different local structures happen inside the protein. Now remember that secondary structure is all about local structure. So how does different local regions in the protein look like? So mainly there can be 3 types of structures, local structures. One is the alpha helix, which looks like a DNA shaped thing. One is the beta sheet, which literally looks like a sheet. And the other one is the proline. So proline is something which is actually not well formed. So it's an ill formed protein, what should I say, protein region. So these 3 structures, these 3 classification of secondary structure is part of the protein Q3 secondary structure genre. Now there is another new class of protein secondary structure which is called Q8. Now Q8 is just basically a broader version of Q3. That is why I'm not going there, but Q8 is there. So in Q8 there are 8 types of secondary structure, and basically those 8 types are subtypes of alpha helix, beta sheet, and proline. Okay, so just to demonstrate that secondary structure is indeed all about local structure. So suppose this is a protein sequence, and in this protein sequence here we have the alpha helix. So this part, this primary sequence is forming an alpha helix structure. This primary sequence is forming, say, beta sheet structure, and this primary sequence is forming, say, another alpha helix structure. Now, for demonstration purpose, I have only written 5 to 6 residues here, but in reality, your secondary structures will be formed using 10 to 20 amino acids. So the regions will be around 10 to 20 amino acid long for the secondary structure. Now, one thing I want to mention here is that functional protein, which actually stays in your body and functions and does different things, cannot really stay in the simple primary structure or the simple secondary structure. It is extremely rare. They cannot stay in this particular structure. So what do? In reality, they form some other kind of structure which is known as tertiary structure. So tertiary structure is a folding pattern, like how the protein actually folds in the actual 3D space. So it's the entire global view of the protein. So previously in secondary structure, we saw the local view, and now we are looking at the global view of the entire protein. So the protein folds into some 3D shape, and then it can function. So it's a stable version of protein. And there are globular structure which is very compact, and then there are sparse structure like fibrous structure as well. These are two major types of tertiary structures. Now, another thing I want to mention is the residue-residue contact map. Now, what is residue-residue contact map? Now, how the protein tertiary structure will fold depends on the primary structure of protein, which is the sequence of amino acid itself. Number two, how the local regions are appearing, that means the secondary structure of the protein, and it also depends on the residue-residue contact map. So what is that? Suppose we have a very small protein sequence which is not real. This is just an example where we have 6 residues. So it's a 6 length, 6 amino acid protein sequence. So what we can do is we can actually see how far each amino acid is from the other. So how far is the first amino acid in the protein chain from the second amino acid? How far is the first amino acid in the protein chain from the third amino acid? And so on and so forth. We can do that, right? Now, obviously, you may be thinking that, okay, if we have amino acid 1, then amino acid 2, then amino acid 3, then amino acid 4 in the primary sequence, then amino acid 1 should be close to amino acid 2, and amino acid 2 should be close to amino acid 3. But that is not the case in reality. In reality, the protein 3D structure can fold in various ways, such that maybe the amino acid 1 is very close to amino acid 6, but amino acid 1 is not so close to amino acid 3 or 4 or 5. This can happen in real life, and it happens a lot. Okay, that is why these 3D foldings look so complex. Now, you can see ones and zeros here. So you can see a 1 right here. That means amino acid 2 and amino acid 1 in the primary sequence are actually very close to each other. So there is a threshold of closeness. I do not want to go there. I just want to emphasize on the matrix itself. But if the amino acid pair is not that close, for example, amino acid 4, amino acid 2 is not that close, then we are going to put a 0 here. So that is what residue-residue contact map is all about. So it's a symmetric matrix because obviously this part, it will be the same as this part. That means the distance between 1 and 2 is the same as distance between 1 and 2 right here. So if this is 1, this will also be 1. So we do not really put anything right here. Okay, the final thing, the final boss, I would say, is the quaternary structure. Now, all proteins do not form quaternary structure. There are few proteins that form this, and it helps them to do their functions better. So quaternary structure basically interaction of subunits of proteins. So multiple proteins, when they interact together, that is called quaternary structure. For example, one of my favorite proteins is hemoglobin. So hemoglobin is a protein that carries oxygen from our blood, right? So it's very crucial for our human body. Now, it is made up of 4 polypeptide subunits. They come together and then they perform the oxygen carrying function in the blood. So that is called quaternary structure. So these are the 4 fundamental structures of protein: primary, secondary, tertiary, and quaternary. So there is one other thing I want to mention, which is known as post-translational modification, or PTM in short. So what is PTM? So obviously we know that DNA transcribes into mRNA, or mRNA translates into protein, and then protein forms some kind of tertiary or quaternary structure. But then there is some other modifications as well. You can see some external chemical molecules or some external radicals are actually joining with the protein chains. That can happen, and that is known as PTM, or post-translational modification. And it has a lot of implications. So if PTM happens, then it sort of dictates where the protein will actually function, what will be the structure of the protein, like how it will fold. It can also alter the function of the protein. It can regulate the activity of how the protein does its functions, and it can also regulate which molecules the protein will actually bind to. So these things can happen. And since, this modification can change proteins so radically, it actually helps to increase the number of proteins even more. That is the interesting thing. So previously we talked about alternative splicing. Just to remind you, right here, this one. So because of this, number of proteins is far greater than number of genes. Another reason number of proteins is far greater than number of genes is because of the PTM. So some examples of PTM can be these 4, 5. Obviously there are much more, but these are some major modifications like glycosylation. It adds the sugar chains. Then phosphorylation, it adds phosphate groups. Then there is lipidation. It adds lipid groups, and then the protein can come into the membrane and perform the functions. And then there are also acetylation, methylation, etc., which are epigenetic modification. So that is all about the proteins. In the next tutorial, I'm going to talk about DNA mutation. Now, the reason I am talking about DNA mutation so late is because you need some knowledge about protein in order to talk about DNA mutations. That is why. So hope to see you in the next tutorial, and I will give the link of the next lecture in the description. Thank you very much.